So this thing, can you click got it from AJ? Hello, YouTube. Welcome to the Water and Wetlands and Watershed Seminar. We're going to record to the cloud. We're going to let everybody into the room and get started in just a moment. So let's get this. Okay, got it. All right, hello, YouTube. Hello, everyone in class and everybody on Zoom. So today is our next Water, Wetlands, and Watersheds seminar for spring 2024. And Aaron, if you excuse me, I always like to ask the students what they remember from last time, just like jog their brains a little bit. So helps me remember as well. So one or two things that we learned last week in seminar.
There's like a pretty huge decline in wetlands in the Midwest. It's like 50 to 90%.
Yeah, so we heard from Sarah McMillan, Dr. Sarah McMillan, right? We heard about all these drain tiles in the Midwest and the impacts on the wetland biogeochemical cycle. What else? One more thing. Yeah.
Like the specific plant or vegetation should be looked at when planting it in a restorative wetland type situation, because each plant has different functions.
Right, so she's looked at these different kind of vegetation, and if you plant this one over here in that kind of wetland and that one over here in this kind of wetland, you get different outcomes in greenhouse gas emissions and nutrient cycles. It was awesome. It was a great talk. You should check it out, Erin, one time. I know you probably know her work. Yeah. All right, but without further ado, today's speaker is Dr. Erin Hotchkiss. So Dr. Hotchkiss is an associate professor in the Department of Biological Sciences. She's also a faculty affiliate in the Global Change Center at Virginia Tech. She received her PhD in ecology at the University of Wyoming in 2013. And after that, she was a postdoctoral fellow at Umeå University in Sweden. Umeå, and also at the Université de Quebec à Montreal in Canada. In 2016, Dr. Hotchkiss received the Heinz Award for New Investigators from the Society for Freshwater Sciences and the Raymond L. Lindemann Award from the Association of for the Sciences of Limnology and Oceanography, or ASLO. She's currently vice president of SFS and became an ASLO fellow in 2023. So congratulations on all that. Ongoing work in the Hotchkiss Lab, which maybe she'll share with you the nickname for the Hotchkiss Lab. I'm not sure if that's in the talk, but she's exploring how environmental change, land-water interactions, and ecosystem processes shape the transport, transformation, and fate of carbon, nutrients, and pollutants in fresh waters. So, Erin, Dr. Hotchkiss, the room is yours. Thank you so much for joining us today.
Great. Thank you, David. And thank you so much for the invitation to share our work. So today I'll be talking about how we're characterizing stream carbon dioxide regimes across different ecoregions in the United States. The core of my work, and if I can get slides to advance, here we go. Yes, okay, so the core of my work will be sharing out from a collaboration that's highlighted with a picture and names of folks on the left of the screen. This is a project funded by the National Science Foundation's Macro Systems Project, where we're linking land to water transport and stream carbon cycling to inform macro system carbon balance. And I'll put some of this work in context with other past and ongoing collaborations out of my lab group, talking about what we're learning about carbon dioxide in different waterways, from intermittent headwaters to large rivers draining our campus stream. And so we're going to start here. This is a stream down the road from my house, obviously in a different season than we are now. But when I look at this site and when I walk down there and see it across a range of different flow levels and water colors and seasons, I'm often curious about a lot of different things. And I imagine, depending on your interests, you may have different curiosity questions that come with a picture like this. But I'm going to center my curiosity questions with some of the things that I'll be talking about today. So thinking about, first of all, this water is full of organic matter. And so as a biogeochemist, I'm curious about how much carbon is being transferred from land to water, how quickly those inputs can be used by microbes and other consumers in the stream and respired to CO2. How much of that carbon dioxide or CO2 is being emitted from this reach? For those of you that think about gas exchange and see all those bubbles in the picture, you might be wondering, how the heck do we measure K, which is air-water gas exchange and often causes us lots of headaches? And also thinking more broadly about how material flux and the fate of those materials that are coming in from the landscape changes with flow as the stream gets larger and smaller over time. And I ask these questions as a stream ecologist and biogeochemist because we know that carbon moves within and across ecosystem boundaries. And the form and the magnitude and the timing of those inputs will set the stage for biological opportunities in stream and river networks. And so whether that's sediment or annual leaf litter fall or boreal streams that are always rich in organic carbon, understanding where these sources are and how they change over time is still a really emerging field, especially because we don't have great CO2 sensor technology to go in streams yet. So a lot of the data I'll be talking about are from relatively new carbon dioxide sensors that we can install in streams and are just starting to get a peek at some of these high frequency patterns in carbon dioxide across different stream types and over long periods of time. And we care about these inputs, whether they're organic or inorganic, because we know that land is leaky, right? Streams receive a lot of inputs from the surrounding landscape, but rarely are those inputs included in terrestrial budgets. So a lot of the carbon that's counted as stored in terrestrial ecosystems actually leaks into streams, lakes, and rivers, and sustains a lot of the biological processes that I study. And so while it's not quantified often, I'm highlighting a recent modeling study that was led by Sierra Talbot, who was a recent PhD graduate from the University of Notre Dame, where she was curious about how these lateral carbon inputs, in this case dissolved organic carbon, would change across a range of precipitation here on the x-axis, and depending on the temperature, so shown here in the different model simulation colors. This paper also does a really nice job talking about different plant functional types and how that may mitigate the amount of organic matter that's produced on land and makes its way into streams. And so we care about quantifying this lateral carbon transport because it's a big question mark in our freshwater budgets. And so what I'm showing you here on the left is a figure from a paper from 2007 led by John Cole, where they argued that we need to do a better job of considering inland waters as not just passive pipes where what comes in from land goes directly out to the ocean, but also be thinking about what happens along the journey as those inputs from land move through freshwater ecosystems. And we know that freshwater ecosystems, most of them are net emitters of carbon dioxide and methane to the atmosphere. And they also can store a lot of sediment, especially in lakes and reservoirs and floodplains. And so only a small fraction of what's coming in from land then makes its way to its ocean. And we don't really know this land number. So usually we're estimating this input based on what we know about what's exported to the ocean, what we think we know about emissions and storage. We also don't really know in what form the carbon inputs from land are coming in. Are we seeing this as just carbon dioxide that's been respired on land, or is it organic carbon coming in from the landscape that can fuel metabolism and food webs in aquatic ecosystems? And so what I'm showing you here on the left now is a figure from a paper by Drake et al. In 2018. On the x-axis is year, and it starts with this Cole paper that I just talked about that was published in 2007. And it covers about a decade of research on carbon export from land based on what we think we know about emissions and burial and what makes its way to the ocean. And so Cole et al. Estimated about 1.9 petagrams of carbon should be emitted from land. But as more and more publications came about, thinking about different processes and different ecosystems, or adding methane to the picture, or identifying patterns of CO2 or methane emissions in understudied regions or ecosystems, we keep on building up our estimates of how much carbon must be leaking from land to sustain a lot of these emissions that we're measuring from waterways. So every time we update our estimates of emissions from a new place or ecosystem, our estimates of input from land is increasing. So this is a huge mismatch, you can imagine, in the terrestrial and aquatic budgets, which means we need to figure out what's going on and try to put some of these pieces together. And so colleagues and I have been trying to do this. This is a recent paper that was led by Tom Batin, where we were putting together the picture of how much productivity and respiration is happening on land, including soils and wetlands, and what is making its way into waterways, both in terms of organic and inorganic carbon, how much is being lost to the atmosphere or buried relative to export to the ocean. And a lot of my work focuses not just on these fluxes, but the so what for the biology and ecology of streams once carbon makes its way into the system. And so I'll be talking about these terms next, and I'm going to define them on the next slide. So here's how we think about the metabolic processes in streams. So on the x-axis is gross primary production or photosynthesis. And on the y-axis is ecosystem respiration or respiration of everything living in the stream, both photosynthesizers and consumers. This line is the one-to-one line, and every point that's on or above the one-to-one line has respiration that's higher than photosynthesis. And so this is a case where we talk about those external inputs from land subsidizing respiration and creating more CO2 in these ecosystems. And we think this changes over space too, but we don't have a lot of network measurements of these processes to go along with carbon dioxide emissions or export. So this is a framework that colleagues and I proposed about a decade ago now, where we were identifying how inputs from land likely decline as we move from small headwater streams to large rivers. The metabolic balance of those systems also shifts from small streams to large rivers. And thus, the role of leaky soils versus stream metabolism and contributing to CO2 emissions should also change as we move across the landscape. Again, this is a generalized stream model based on mashing together data that we had for metabolism and CO2. And so what we're working on now is actually making some of these measurements concomitantly at sites so we can put these pieces together in a more effective way. So when we think about the role of streams in landscapes, not only are they almost always heterotrophic, so this is what I talked about with respiration almost always being higher than photosynthesis, supported by these inputs from land. Most ecosystems outside of streams don't see the same subsidy in the amount of respiration that's happening in excess of photosynthesis. So what I'm showing you here is net ecosystem production. When you have a negative value, your respiration is subsidized by sources other than internal photosynthesis. And when you have a positive value, you're storing more carbon than you're respiring. So we typically think of lands as storing more carbon than they're respiring, and streams are an extreme and being really subsidized by these external inputs for their biogeochemistry. And we know that per unit area, and again, this is streams cover a much smaller surface than terrestrial ecosystems, but when we compare these fluxes on a meter squared per year basis, again, streams are respiring much more carbon than terrestrial ecosystems seem to be storing. And so these are data from global streams and rivers that were put together by some collaborators and I, during a COVID project that we're now wrapping up into a publication, just thinking about, how can we put these pieces together to really understand what's happening at a global scale. And we know that when we want to understand, in addition, global estimates are great, but they don't tell us a lot about mechanisms because we don't see the patterns like I'm showing here. And the patterns are what I'm going to talk more about today. So I wanted to put my work in a larger scale context, put it in the context of other ecosystems, but also zoom in to say, okay, so, what's happening at unique sites? What are the patterns that we see? And what can we learn from those patterns as we have more and more sensors in the environment that we can track some of these processes over space and time? So here I'm showing you oxygen data, multiple years of oxygen data. So from 2012 to 2015, and oxygen saturation on the y-axis. And these are two sites with really different, what we would call CO2 regimes or oxygen regimes. And we know that the oxygen saturation in streams can change either when it's enhanced due to higher light and nutrients driving photosynthesis, or when it's lower due to higher respiration or grazing of algae or flow-induced disturbances. And so just from these two sites, we can already expect that the signals that we see in different streams are going to be quite different. And the metabolism that's driving those signals is also going to be different. And so if we compare the metabolism here, gross primary production is shown as a positive value, and ecosystem respiration is a negative because it's consuming oxygen. For these two sites, the patterns of metabolism looks really different. This site is much more productive. Primary production is driving respiration. We often see these two processes mirror each other, especially when primary production is important in fueling respiration in the sites. Whereas this site, metabolism is much more constrained, much lower gross primary production, and we would expect for respiration to be a bit detached from photosynthesis in this site, and likely more subsidized by external inputs. And so from data like these, so I've shown you just two sites with very different oxygen regimes and metabolic regimes. We're starting to put together pieces of, characteristic signals of different sites, and we use regimes a lot to talk about those characteristic signals. And so what I'll talk about today in the context of land being leaky, freshwater ecosystems being emitters and processors of carbon, and trying to understand signals of different ecosystems from high frequency data, we're asking to what extent do stream CO2 dynamics or regimes reflect terrestrial inputs, as well as hydrologic regimes and metabolic regimes. And so I wanted to briefly share out two examples of what these regimes might look like across different sites for two different processes. On the left is a cluster analysis of different hydrologic regimes. The authors use this to talk about future predictions for stream flow, but I think this does a nice job of showing how, depending on where you are in the US, you might expect to see really different patterns in stream flow that are going to govern when materials are coming in from the landscape, when critters in the stream are getting stressed from too much flow. And so that can look like intermittent streams that blink on and off seasonally, sites that are governed by a really high snowmelt period, or sites that have kind of more of a winter wet up period, like where I'm working here in Virginia. And so just like the hydrology has regimes that are characteristic of climate and geomorphology, streams seem to have productivity regimes. So this is a paper by Phil Savoy and colleagues where they did a cluster analysis on photosynthesis patterns across different sites. Day of year is on the x-axis and gross primary production is on the y. And this is showing 4 of the common productivity patterns that we see in streams across the US. Aseasonality, which was similar to that example that I showed in the first 2 stream regime plots in a prior slide. Summer peak may be what a lot of people think about, especially if you think about productivity regimes with a terrestrial mindset. Summer declines happen quite often, and those are associated with these spring peaks in productivity. And we're getting ready to see that happen here where I live. So the leaves haven't leafed out yet, right? So the streams are still getting a lot of sun. It's getting warmer. And so we often have these spring pulses of photosynthesis that then fuel, either ongoing productivity, but at a little bit lower rate throughout the summer, or we just see this spring peak. And then as soon as the leaves come out and shade out the stream, we're kind of done with our peak photosynthesis for the year.
Erin, can I jump in for a second? Just to
Yeah.
kind of query the students' brains for a second. So maybe it's a spoiler, but these kind of patterns on the right that she's showing about stream metabolism, do you think they're going to have the same sort of like spatial consistency that the rain, like the other patterns about the stream flow are about rain and climate and all that? I don't know the answer. Maybe it's a spoiler, but do you guys think that those patterns on the right about metabolism are going to have the same sort of geographic consistency as the patterns on the left? Maybe just think about it a little bit. And what else might be the things that determine which, and I'm sorry, Erin, but I gotta like play with you guys a little bit. Yeah. All right. So, okay, we'll keep
Make sure everyone's awake.
and if not, you better ask her at the end. Okay, go ahead.
Yeah,
Sorry.
no, and so I'll say that yes, if we were in kind of equal pristine landscapes for all of these sites, right? And so, you know, the initial data that I'll talk about for CO2 is relatively pristine landscapes, and then I'll give some tidbits from a local campus stream where we see things, you know, kind of change because the landscape is not, you know, this nice forested system. It's concrete and parking lots and buildings. So to put all these pieces together, we really need to take a meta-ecosystem perspective. And so for those of you not familiar with this term, I wanted to make sure that I define it before I use it moving forward. And so when we're talking about meta-ecosystems, this is considering not a single ecosystem at a time, so only a stream or only a wetland or only a forest, but looking at a set of ecosystems because they're connected by spatial flows of energy, materials, and organisms across those boundaries that we impose as scientists. And those spatial flows across ecosystem boundaries end up being really important for a lot of the things that we care about in terms of water quality and ecology. And so the project that I'll talk mostly about today is asking what we can learn from moving beyond these isolated ecosystem measurements to characterize carbon fluxes and fate at the meta-ecosystem scale. And so these two images are cartoons from the grant that was funded, and the numbers correspond to the same processes that we're tracking. So we're thinking about terrestrial carbon metabolism, storage, and emissions. We're thinking about those connections between land and water, both kind of overland export when it happens, as well as subsurface connections that are, you know, putting in different water and chemicals into the stream ecosystems. We're thinking about the metabolic processes in the streams that I've talked about already, as well as the emission losses that happen before that water goes downstream and fuels emissions or metabolism in downstream ecosystems. And so we're working at 5 National Ecological Observatory or NEON stream sites. And we chose these 5 sites because they have the closest terrestrial monitoring sites that are also NEON long-term sites as well. And I'll talk about the sites in a minute, but I just wanted to give you an idea of the different types of measurements that NEON is making and the additional measurements that our project is making to overlay with those publicly available data. So NEON is measuring terrestrial net carbon exchange fluxes and also soil carbon and respiration data, in addition to lots of other really cool terrestrial data sets that they're providing to our scientific community. They also measure groundwater biogeochemistry, so they have wells that are installed adjacent to their core stream sites, and our project has increased the frequency of sampling at some of those wells for both carbon dioxide and dissolved ions. NEON is also monitoring stream biogeochemistry. They have sensors installed in streams, and they also take measurements across their core stream sites over space and time. And so we have data on in-stream biogeochemistry, including stream metabolism and hydrology. And we've also installed aquatic CO2 sensors at these 5 NEON sites. We managed to get a couple years worth of data before the CO2 sensors were recalled by the manufacturer. And so there's some gaps in our data that I'll talk about. But again, this is just to stress that we still need tech to catch up with the questions that we have in some of these ecosystems. And so the 5 core sites that I'll share some CO2 regime data from today and link up to some other work that we're doing more broadly are at Caribou Creek in Alaska, Martha Creek in Washington, Como Creek in Colorado, Kings Creek in Kansas, and Walker Branch in Tennessee. And I'll talk a bit more about these sites and the ecoregions that they occupy as I talk through some of the initial carbon dioxide data, but just wanted you to kind of see the sites across the North American landscape. Maybe think about some of those hydrologic regimes that I showed you in the initial slide and how we would expect the hydrology to be quite different across these sites and influence our carbon dioxide data in different ways. So we're going to go right into some discharge and CO2 data, and then we'll talk about what we're learning from it. And I'll talk through each site individually before we put all of the data together to see how the regimes compare to each other. So we're going to start up in Alaska. This is a boreal subarctic site with patchy permafrost extent. The landscape is woody wetlands, hardwoods, and shrubs, and the site is characterized by cold winters and a pretty big snowmelt pulse. The sensors were removed over winter because we didn't want the ice to crush the sensors, and so you'll see some gaps in the CO2 data due to that. But we're seeing the signal of a snowmelt pulse and then increased productivity that's driving this annual pattern in CO2. And so the data that I'll be showing for these sites has day of year across the x-axis. So often there are multiple years embedded in these plots, but just showing across a single year to think about these regimes and what we can learn from them. The top plot will be discharge of the stream in meters cubed per second, and the bottom plot is CO2 from our in-stream sensors measured in ppm. So you can see this big snowmelt pulse that happens. Again, this is the subset of the year where we have data for the Boreal site. And from some work that colleagues are doing to unpack the metabolic patterns in these sites, we're seeing, a strong role of metabolism kind of picking up during the summer period. And we can see that in the variability. If we zoom in and look at the diel changes in carbon dioxide, it's decreasing right during the day, increasing at night, following some of those metabolic patterns and forcings of carbon dioxide at these smaller timescales.
Aaron, could you move like 2 inches closer to the mic, because it's kind of coming in and out, and sometimes it's super clear, and I'm not sure if it's network or if it's hardware.
Yeah, no, thank you for asking, and let me know if that process doesn't help.
That's better. That's better.
Okay,
yeah.
cool. I will not sit back so comfortably then. Okay.
All right.
All right, so next up is Martha Creek. This is our Pacific Northwest site. It's surrounded by old growth coniferous forests and has cool, really wet winters and warm, pretty dry summers. So we have kind of a strong wet, dry season here that contrasts with some of our other sites. And here we see carbon dioxide build up over summer and fall, and that's disrupted as soon as the winter wet season turns on. So again, discharge of the stream is on the top. Carbon dioxide is on the bottom. Some of these scatter points are signals of dying sensors. And we do see, again, this buildup and then decline. And a lot of dilution is happening at this site during the higher flow periods. So we're not seeing a metabolic signal of CO2. We're seeing more of a rainfall signal of CO2. All right, Como Creek, Southern Rocky Mountains, conifer and shrub landscape, really, really big snowmelt pulse here. I mean, we do have sensors left in over winter, so we can show you some under ice patterns that I think are pretty cool. So we are seeing CO2 buildup under ice. It's diluted by the snowmelt pulse and then stays low during the growing season at the site. It's relatively dry. And also very shaded. So you can see a little bit of dappled light in these phenocam photos showing different seasons. But here I just want to stress this under ice buildup that we see, this crash of carbon dioxide that happens with the snowmelt pulse, relatively low carbon dioxide throughout the growing season, and then buildup of that carbon dioxide under ice again. This is the only site where we see an under ice signal. We may have seen it in Cary if we could leave the sensors in Alaska over the winter, but we will not know. Second to last is Kings Creek. This is Continental Prairie. It's a mixed agricultural pasture and tall grass prairie site. It's managed a little bit for grazing bison and has some fire management in the landscape, but is still relatively pristine, as are all of the neon sites. It has cold, dry summers, or cold, dry winters and warm, wet summers. And it does go dry. So I don't know how well y'all can see this from the slides, but here, especially during the winter extremes, the stream bed is totally dry a lot of the time. And we see that signal in both the discharge and carbon dioxide. And we see these extreme CO2 values much higher than any of our other sites that are associated with these wet up pulses. So as the stream that was kind of a series of dry stream bed or pools reconnects, we see some of that really high carbon dioxide make its way into the waterways and likely emitted pretty rapidly before it comes downstream. All right, and Walker Branch is closer to where I am, and I'll share some other case studies from this region moving forward. This is a deciduous temperate forest. It has cool, mild winters and hot, humid summers. Really dominated by leaf fall pulses, and is also one of those sites where we see that spring shoulder season. So most of the productivity happens in spring before leaf out. when the algae can actually get some sunlight. And here we see, again, relatively wet winters into dry summers, a buildup of CO2 during the summer. We think it's relatively low during these shoulder seasons, but these were also seasons where we've had problems with sensors, and we're hoping to fill in those gaps through our last data collection this year. So I want to put these all together for a moment before we kind of talk a little bit more about the connections that we're making. Just to stress that these are 5 sites that we picked because of strategy based on where NEON was sampling, but occupy different hydrologic regimes, are in very different ecoregions in terms of climate and contributing landscape. And we're seeing very, very different CO2 regimes across multiple years at these sites, depending on what some of these key drivers are, both on the landscape and in the streams themselves. And so next I want to show you these patterns of CO2 and discharge related to each other. Often we talk about this as CQ, or concentration discharge patterns in these streams. This work was led by Frances Anucci, who was a master's student at Fairbanks, and then worked with me as a data scientist after she completed her master's. And on the left-hand side of the screen, I'm showing you these flow CO2 relationships for 2 of the sites, and I'll build on the other sites as we talk through them. So now instead of high frequency data, I'm showing median discharge on the x-axis, and carbon dioxide is on the y. These data are color ramped by water temperature in degrees Celsius, and the axes and color ramps are all different among the sites. But what we see for these 2 first sites here, Boreal forest, as well as the temperate deciduous site in Tennessee, is lots of variability at low discharge, right? So when the stream flow is low, we often occupy the full range of CO2 values at this site, but it's changing seasonally. And when we do see these high flow periods or high flow events, these sites pick up CO2 from the landscape. And so we're seeing higher flows bringing in more carbon dioxide to these streams. And at these 2 sites, based on our understanding of metabolism at both of these sites, the low flow and low carbon dioxide periods down here in both of these plots are also times that have higher gross primary production or higher photosynthesis in the stream, which is going to be removing carbon dioxide from the water column. The next 2 sites with kind of similar patterns as we're looking at discharge and carbon dioxide. Again, the axes are the same, discharge on the X, carbon dioxide on the Y. And now we have the temperate rainforest site and the Southern Rocky Mountain site. At these 2 sites, we see higher flows diluting carbon dioxide. So again, these low flows occupy the full carbon dioxide range, but those high flow extremes are coming with lower carbon dioxide in the water. And our non-perennial site is kind of somewhere in between these 2, right? Misbehaving, but has some really interesting patterns. And I also just want to note, if you compare the y-axis of this non-perennial site in the tall grass prairie to the y-axis on these other sites, it also has the highest carbon dioxide by far. And so there's been a lot of work to really increase our understanding of non-perennial streams and their contributions to biogeochemistry, and carbon dioxide pulses that come with these non-perennial streams wetting up and drying down, I think are a really important aspect of understanding what's happening on the landscape. So just kind of big picture, putting things together before I tell a couple of case studies to put this work in broader context. We found distinct CO2 regimes among the 5 streams that we're studying in different US ecoregions. These CO2 sources, whether from shallow soil, deeper groundwater, or in-stream metabolic processes, as well as the form of carbon inputs and those input flow paths, is differing substantially among streams and seasons. We're finding that metabolic regimes and terrestrial inputs are really strongly linked to CO2 patterns at the temperate deciduous and boreal stream site that we're studying more closely. And these hydrologic regimes are driving CO2, especially these dilution events that come with higher flow days in our Rocky Mountain temperate rainforest, and also our non-perennial prairie stream. And so this project is, we're wrapping up our final year of data collection. We've got a lot of cool work that's ongoing that I won't have a chance to talk about today in great detail, although I'll share a little bit of one of these projects if I have time, where folks at University of New Hampshire, led by Asif Abir, who's a PhD student working with Will Woolheim, is looking at soil and groundwater data to build stream network CO2 models from the data that we're collecting in the analyses I shared. Kristen Olson, Francis Yanucci, and Jay Jones are thinking about the permafrost extent across a range of streams in the Boreal zone, linking that with terrestrial net exchange and seasonal dynamics to understand those boreal streams. Hannah Conroy, working with David Buttman at the University of Washington, is also thinking about network scale patterns in stream CO2 emissions, but at different flow stages. So thinking about those wet-dry periods or wet up of the non-perennial stream site. And then some ongoing collaboration between Francis and I is looking at how we can integrate these high frequency oxygen, CO2, and stream discharge data to look at these controls on carbon dioxide at different time scales. So ranging from diel to event scale to seasonal up to the multi-annual regimes that I shared today. And so what's missing from a lot of these single site perspectives, even though we need them to start building our understanding of what the CO2 signals in different streams are, is spatial variability and also landscape context. And so we do have a non-perennial stream, but we're missing, kind of characterizing any of these contributions of adjacent wetlands or upstream lakes or intermittent segments at a lower stream that we might be monitoring more intensively. And what is notably missing from both this diagram and from the NEON study sites are really intensive human impacts. And so I'll share a little bit of data from a campus stream where we've been studying some of these processes more intensively. So 4 quick case studies. We'll go down to Georgia to talk about how mountain stream corridors with vernal pool wetlands have different CO2 signatures. We'll go back to Blacksburg, where I am, and look at how a mountain stream with non-perennial flow and a stream draining a developed landscape have different carbon dioxide signals. And then we'll go out to Martha Creek again and learn a little bit about the work that Hannah Conroy is leading to think about these network CO2 patterns based off of our NEON data and some of her sampling data that she's been building for some of these sites. All right, so we're going to start with Coweta, Georgia. You may see someone that looks familiar in this picture. So this is work led by Kristen Bretz, as well as some, at the time, undergraduate collaborators, Jonathan Monroe, Alexis Jackson, and Samaya Rahman. All 3 of these students are working on graduate degrees in various places, both in biomedical and ecological engineering. And we looked at these carbon dioxide patterns, again, not at a single site, but at clusters of sites in the broader Coweta watershed. So I'll be showing data from 4 streams. 2 have nearby vernal pool wetlands and 2 do not. And they're all in this same kind of complex of wetlands. So instead of thinking about different biomes, we're now going to see how different are sites that are next to each other that should maybe look similar unless we think about the broader context of those sites. And so this is one of the final figures from this paper that Kristen led that was published in 2021, where for these 4 sites with jargony names that probably don't mean much to folks, but I'll just walk through with the names so you can follow which one I'm talking about. Watershed 5 and Tower Stream were the 2 without vernal pool wetlands, and Old 5 and Ball were the 2 with vernal pool wetlands. And we measured the CO2 emissions at different patches along the stream corridor, both dry beds when the stream was flowing underground, dry wetlands during drier seasons of measurement, hill slope adjacent to the stream itself, and the wetland if it had standing water. And across all of these sites, even when the wetland was dry on the surface, but likely still very saturated below our measurements, we saw much higher carbon dioxide flux here on the x-axis at especially this Ball Creek site that had really large adjacent vernal pool wetlands. And so again, these 4 sites are right next to each other. If we just put a sensor in one, we might have walked away with a really different picture of what's driving carbon dioxide at this site. And so even though getting a single site characterized in a certain ecoregion is going to be an important first step, We can't forget the complexity and the individuality of different sites that may be right next door to each other. All right, next we're going to go back up to Virginia. So this is in the National Forest right outside of town, and work also led by Kristen Bretz as part of her PhD in collaboration with Natalie Murphy, another undergraduate in our lab that has since graduated, and also collaboration with Rooster Snyder, who is shown down here in the bottom picture. So this is an intermittent stream in National Forest outside of town. I'm showing you base flow, mid base flow, high base flow, and moderate storm conditions across a range of sites. And I'll show you oxygen and carbon dioxide from this one stream, Beast Stream, as it's going dry and wetting up again. So here on the left panel is discharge on the x-axis, and carbon dioxide and dissolved oxygen on the y. Dissolved oxygen is the top plot, carbon dioxide is the bottom plot. The carbon dioxide and discharge axes are log transformed, and we see very different responses to stream flow. The CO2 gets diluted with higher flow, but this is also a seasonal pattern. And so the light pink data are associated with August, which is the driest part of the season, also when discharge is the lowest. And what we can see changing over time is essentially the wet up and reconnection of the stream as stream discharge increases through storm pulses and finally reconnects persistently towards the end of December. And so we see these signals from the landscape of not bringing in more carbon dioxide at these higher flows, but pushing out a lot of carbon dioxide from these patchy pools as the stream first starts reconnecting. And we see an inverse pattern between carbon dioxide and dissolved oxygen across these sites. And Kristen has another paper that she's wrapping up that talked about the metabolic patterns that are driving the DO and carbon dioxide correlation. All right, so we're going to move from reference to developed landscapes. We're still going to be in the temperate Appalachian Plateau region of the US, but Walker Branch, which was one of the neon sites that I talked about, this is its land cover, all forest, and the B Stream, also all forest. Most of the sites in Appalachian Plateau are not forested anymore. Most of them are draining really heterogeneous developed landscapes like the one that I'm going to be talking about for Strubles Creek. So Strubles Creek originates in the town of Blacksburg. It's actually underground throughout a lot of the town of Blacksburg and underground through a lot of Virginia Tech's campus. We have an open canopy segment of the stream that's associated with a little bit of pasture land that I'll be talking about some data from today. But just wanted to show the contrast in both stream size. So what I'm showing you here, watershed area, but also land cover, and stressing that we need to get some of these messier landscapes into our data sets. This is a site that's very productive. So this is a single site that really occupies the same range of gross primary production and ecosystem respiration data that I showed for the global data set. Almost always heterotrophic, so respiration is almost always being subsidized by external inputs, but also a very productive stream. So gross primary production occupying a really full range of values that corresponds with what we see from global sites from low to high productivity. And we know that higher stream flows disrupt metabolism at both of these sites. This is work led by Bryn O'Donnell, where we see across a range of discharge here on the x-axis, GPP declining with increased flow, and respiration declining a little bit, but not quite as much. And so we know that primary production is more vulnerable to flow disturbances than respiration. And when we look at carbon dioxide in the stream, I'm showing you a snapshot of carbon dioxide on the upper plot here and oxygen in the lower panel across several days. So we're seeing these diel signals that are associated with biology in the stream. When carbon dioxide is high, oxygen is low. That means respiration is dominating the processes at the site. And when oxygen is high, carbon dioxide is low. This is during the day when photosynthesis is governing what we see. We can also see this kind of wonky disruption in those lovely diel signals. That's a storm. And so the storms disrupt oxygen and carbon dioxide in different ways. And when we look at those patterns adjacent to each other across a multi-year data set that we have for this site, We see, again, a strong correlation between oxygen and carbon dioxide, but that it shifts seasonally with the productivity changes at different sites as well. And when we look at the time series in the way that we did for the neon sites, we see a very flashy hydrology. It's not very seasonal at this site because of all the concrete cover and the buried stream segments. It does wet up again, in the winter, like the more pristine sites do, but it's not as stable hydrologically as more of the pristine sites where a lot of our more intensively studied data are coming from. And we see in general that these frequent flashy high flow periods dilute stream carbon dioxide and disrupt these metabolic signals. And I didn't plot the medium discharge like I showed for the other sites because I wanted us to be able to see that each one of these storms, which is what we're seeing with these loops that are showing up in the discharge and CO2 data, push the carbon dioxide across quite a range of values as you're increasing discharge and then decreasing discharge across a single storm event. All right, keeping an eye on time.
Including questions, 5 minutes total.
Okay, all right. It's not an hour?
50 minutes, sorry.
Okay, all right. That was not what I had in the email, but no worries.
Yep.
All right, so I'm going to unfortunately have to skip this case study that was Hannah's work. It was recently published in the Journal of Geophysical Research Biogeosciences. She's doing some really cool network scale analyses of how both discharge and carbon dioxide is changing across a stream network and using some really cool modeling to map network carbon dioxide dynamics. So what we're learning more broadly is that we can't only study biology or hydrology or assume that landscapes aren't leaky if we want to understand carbon dioxide dynamics in streams. We're seeing contrasting signals across different ecoregions that I highlighted from the 5 core neon sites. We're also seeing variability within stream networks. This was the paper that was led by Hannah that I mentioned, and how these signals in nearby streams may actually be higher than what we see at a single site in terms of variability over time. But the landscape context also matters. And so when we're studying these intermittent waterways in our headwater networks or urban streams, we need to be thinking about how the dynamic connections with CO2 sources are changing and how altered regimes and developed landscapes are going to be influencing processes that we care about. So I'll stop there for questions given time, and would be happy to take questions and chat with folks after the talk if you'd like to set up a time to chat.
Thanks, Darren. Let's give a round of applause to Dr. Hotchkiss. And I'll check the Zoom and YouTube for questions, but we can start in the room with questions for Dr. Hotchkiss.
I'll start it off. I had a question, going back to looking at the different wetland types, their different emissions, and my brain went right to that prairie because I immediately noticed just like how large of a difference the scale was. And I noticed that you didn't talk a lot about vegetation in this, which I guess it makes sense. Like it's a stream. I guess you're not worried about vegetation as much, but do you guys think about that? Because when I think about prairie areas and I think about just because they're grassy areas that may contribute to their productiveness compared to like, well, my eyes, wetlands types, just like mangrove areas, like more forested areas, just like their vegetation has a big deal in this. Do you guys take that into account at all in this? Like, okay, what is this stream surrounded by? Would that then have an influence into its productivity?
Yeah, absolutely. And I think we've thought about that more generally, right? In terms of just, how does the landscape differ? Is it deciduous? Is it, coniferous trees? Is it tall grass prairie? But a next step that I think is going to be really important as we build the network models, especially that we're thinking about for this project, is thinking about the organic contributions that are coming from these different landscapes and how we would expect to maybe see some really rich pulses of organic carbon coming from the tall grass prairie sites, but that carbon would look very different than, coming from fringing wetlands near the Boreal site, for example. And so that's something that I'm really excited to think more about in terms of making those soil and stream connections more explicitly. And some of the modeling that Asif, a PhD student at UNH, is thinking about is really in the soils and thinking about the soil structure and the respiration and how, as you move up through the soil pore space towards the surface, right, we're seeing different carbon dioxide signals that we can use to learn about what's being exported to the stream itself as well. So maybe a bit more than what you were asking for, AJ, but yes, absolutely, right? And we need to do much more of that if we're going to understand what's in the stream. We can't just ignore, put blinders on and say the stream's the stream.
So Aaron, there showed a lot of data about CO2 concentration in the water, and then there was one study in particular more focused on the flux and everything. But I think for folks who maybe aren't aquatic metabolism gurus, like thinking about the flux from streams and wetlands and aquatic systems, like where do we expect both landscape development and climate change to be taking, just on the broadest brush? Like, are we looking at very simple story about increased CO2 and methane efflux from aquatic systems? Is it much more complicated than that? What can we sort of take home about in-stream aquatic metabolism driving larger scale biogeochemical, like global climate change, more or less?
Yeah, I guess I think about that in two ways. One is how we would expect the hydrology to change, right? So increasing periods of drought and more intense storms, I think, are going to disrupt a lot of these patterns, both in terms of what comes in from the landscape to sustain stream processes, and also just what the streams look like, right? And so thinking about these patterns that we see emerging as we compare discharge and carbon dioxide, I would expect that some of those higher intensity storms or longer dry periods may shift some of these patterns over time in ways that we can't quite anticipate, but we hope to be able to make predictions as we're building out these models using these types of relationships. From an in-stream perspective, I think likely we will see, that's tough, because if nutrients are increasing due to changes in land use, we may see lower carbon dioxide if it's fueling primary production. But I would guess in general, we would expect to see streams and wetlands being increased emitters across the landscape, both of carbon dioxide and methane. I will say in Coweta, where I shared some of the vernal pool associations with stream carbon dioxide emissions, we experimentally warmed a stream for a couple of years, and we didn't see a metabolic signal in response to that. So some things like leaf litter breakdown or invertebrate dynamics changed in response to warming, but metabolism itself didn't change in response to that warming signal. And so there may be some functional redundancy in the microbial population that doesn't respond in as extreme ways to some of the climate change forcing that we're seeing in ways that other organisms likely will, right? And so metabolism is mostly done by microbes. And so I think some of the climate responses that we might expect will be different than for macroinvertebrates and fishes and plants, for example.
I think it's helpful to think about the climate impacts on the hydrological cycle, maybe as a stronger and much more structuring effect than some warming, like in an otherwise stable system. Thank you.
Yeah.
All right, last chance. Any more questions for Dr. Hotchkiss? All right, Aaron, thank you very much. Apologies for the sort of pulling the rug out from under you at the end.
No worries.
Really appreciate being here.
Yeah,
and we will see, so it is spring break next week, so don't come next week, and we'll see you the following Wednesday in this room. We have an in-person speaker, I'm pretty sure. I'll check and I'll let you know, but thanks very much. Thank you, Aaron.
Thank you.
I'll stop the recording.